Welcome to the Deep Dive. We're your shortcut to getting well-informed, taking complex stuff from serious sources, and breaking it down. Today, we are really getting into it. Oxidative phosphorylation. It sounds complicated, and, well, it is, but it's also fundamental to how life gets energy. We're using Chapter 19 of Lehninger Principles of Biochemistry, the 8th edition, as our guide. So if you're aiming to grasp the molecular details, the pathways, the energy dynamics, you're definitely in the right place. Our goal for you: to cut through the density, find those aha moments, and really appreciate the elegance of how your cells power everything.
Yeah, and what's just incredible is how central this one process is. I mean, oxidative phosphorylation, it makes the vast majority of ATP for almost all organisms that aren't photosynthetic. It's more than just energy currency. It's this intricate molecular dance rooted in a really profound evolutionary story. It basically underpins aerobic life.
Okay, so to get that story, we need to start with the mitochondrion, the powerhouse, right? But
Sure.
that term feels a bit simple now.
It really does undersell it, because beyond ATP, these organelles, they're involved in generating heat, making steroid hormones, even triggering programmed cell death, apoptosis. It's much more versatile than just an energy factory.
And that versatility, you're saying it links back to how they originated.
Exactly. If you connect it to the bigger picture, think about this. Over 1.4 billion years ago, maybe 1.45, this symbiosis happened. A bacterium moved into a primitive eukaryotic cell. And that's not just history. That bacterial heritage, you see it everywhere in the mitochondrion today. Its own DNA, its structure, how it works, it screams bacterial origin.
Wow, that's kind of mind-blowing. We're carrying ancient bacteria inside us. Okay, so how does this internal powerhouse actually do it? How does it make the energy? What's the core idea?
Well, at its heart, it's all about energy transfer, but highly organized. Think of electrons flowing downhill, energetically speaking, like water over waterfalls. This flow releases energy. But here's the genius part. That energy isn't just lost as heat. No, it's immediately used to pump protons, positively charged hydrogen ions, uphill across a membrane.
Creating a separation of charge, like building up pressure?
Precisely. You create this stored energy gradient. It's like charging a battery. This is the core of Peter Mitchell's chemiosmotic theory. Honestly, one of the great unifying ideas in modern biology.
So capture electron energy, create a proton imbalance. Got it? And the mitochondrion structure is built for this.
Absolutely. You've got the outer membrane, which is quite porous, but then the inner membrane, that's the key barrier. It's very selective, impermeable to most things.
And that's where the action is, the electron carriers and the ATP making machine.
That's right. The inner membrane holds the electron transport chain proteins and the ATP synthase. And inside that, in the matrix, you have the enzymes breaking down fuel, like from the citric acid cycle and fatty acid oxidation.
Okay, so these electrons, they get passed along. What's carrying them?
It's like a parade of specialized carriers embedded in that inner membrane. You've got molecules based on nicotinamide, like NADH. Others use flavins, FAD. Then there are cytochromes with iron sulfur proteins. But a really interesting one is ubiquinone, coenzyme Q, some call it. It's different because it's lipid-soluble. It can move freely within the membrane.
Oh, okay, so it's not fixed in place.
Exactly. It acts as this mobile shuttle. And crucially, it can carry both electrons and protons. That makes it central to linking the electron flow part to the proton pumping part.
And scientists figured out the exact order of this handoff. How?
Through some really clever experiments, they looked at the natural tendency of electrons to flow, measured reduction potentials. They also used inhibitors, like molecular roadblocks.
Ah, so they'd block the chain at a certain point and see what piled up behind it?
Precisely. Using things like rotenone or cyanide, they could pinpoint where electrons were getting stuck, and that helped map out the sequence, step by step.
Okay, so these carriers, they're organized, not just floating around randomly.
Oh, definitely organized. They form these large assemblies, 4 major multi-protein complexes. We call them complex 1, 2, 3, and 4, all embedded in that inner membrane. Often, complexes 1, 3, and 4 even cluster together into what's called a respiracum, like a little assembly line, maybe for efficiency. Complex 2 is a bit different, sometimes floats more freely, and it's unique. It's actually part of the citric acid cycle too, the only membrane-bound enzyme from that cycle.
Right, so let's talk energy conservation. When NADH passes 2 electrons all the way to oxygen, you said it releases a lot of energy. How much gets captured?
It's quite efficient. Transferring those 2 electrons from NADH to oxygen is highly exergonic, releases about 220 kilojoules per mole. A good chunk of that energy is captured by pumping protons. For NADH, about 10 protons get pumped out for that pair of electrons. This creates the proton motor force you mentioned earlier, It has two parts, a chemical difference, the pH gradient, and an electrical difference across the membrane. It's a powerful way to store energy.
10 protons per NADH. That seems like a lot of stored potential.
It is. It represents a significant portion of that initial 220 kJ/mole being conserved.
But this whole process, using oxygen, dealing with high-energy electrons, are there any, downsides, any dangerous byproducts?
Yes. That's an important point. It's not perfectly clean. A small fraction of the oxygen, maybe 0.2%, up to 2%, can get partially reduced, forming what we call reactive oxygen species, or ROS.
ROS, like superoxide, hydrogen peroxide,
Yep.
the stuff that can damage cells.
Exactly. Those are the main ones. And yes, they can damage DNA, proteins, lipids if their levels get too high.
But presumably the cell has ways to cope.
Oh, absolutely. We have dedicated defense systems, enzymes like superoxide dismutase, glutathione peroxidase. They're constantly working to neutralize these ROS. And here's a fascinating twist. It turns out low levels of ROS aren't necessarily bad. The cell actually uses them sometimes as signals.
Signals? How?
Yeah, like signaling molecules to help coordinate metabolism or trigger protective responses. So life has even co-opted this potentially damaging byproduct for communication. It's quite clever.
Okay, so we've built up this proton gradient, this stored energy. Now the payoff, making ATP. How does that proton gradient get converted into chemical energy in ATP?
This is where the coupling comes in. Electron transport and ATP synthesis are tightly linked. You really don't get one without the other under normal conditions. It's an obligate coupling.
How do we know they're so tightly coupled?
Again, experiments with inhibitors are key. If you add something like oligomycin, which blocks the ATP synthase enzyme, electron transport also grinds to a halt.
Okay.
Conversely, there are chemicals called uncouplers, like DNP244 dinitrophenol. They essentially make the membrane leaky to protons.
So the protons just flow back without making ATP.
Exactly. They bypass ATP synthase. When that happens, electron transport can race ahead because the proton gradient doesn't build up, but all that energy is just released as heat. No ATP gets made. And maybe the most convincing proof, scientists could artificially create a proton gradient across a membrane in a test tube, add ATP synthase in its substrates, and boom, ATP was made. No electron transport needed, just the gradient.
Wow. Direct mechanical link. So what's the machine doing the work? The ATP synthase?
Yes, ATP synthase, also called complex V. It really is a splendid molecular machine. It has two main components, F1 and Pho.
F1 and Pho.
Right. F1 is the part that sticks out into the mitochondrial matrix. It's the catalytic part, where ATP is actually made. Pho is the base, embedded in the inner membrane, and it forms the channel or pore that protons flow through.
Okay, so protons flow through Pho, and that drives F1 to make ATP. How?
Well, this is where Paul Boyer's binding change mechanism comes in. It's brilliant. He realized that the actual chemical step of forming the phosphanhydride bond of ATP on the enzyme surface, it doesn't actually require much energy input. It's readily reversible.
Wait, forming ATP is easy for the enzyme? Then what needs the energy?
Getting the newly formed ATP off the enzyme. The enzyme binds ATP very tightly. The energy from the proton flow isn't used to make the ATP bond itself, but rather to induce a conformational change in the F1 part that forces it to release the ATP it just made.
So the protons push the ATP out. That's wild.
Essentially, yes. It drives the release step, which then allows ADP and phosphate to bind for the next round.
And this involves actual rotation, like a motor.
Exactly. It's called rotational catalysis.
Yeah.
As protons flow through the Pho channel, likely through a ring of subunits called the C ring, they cause this ring and an attached central stalk, the gamma subunit, to physically rotate relative to the F1 headpiece.
A spinning motor inside the mitochondrion.
A tiny molecular turbine, yes. This rotation of the gamma stalk inside the F1 head forces the three catalytic sites in F1 to cycle through different shapes or conformations.
What kind of shapes?
Typically described as three states. One that binds ADP and phosphate, loose or L. One that catalyzes the ADP formation and binds it tightly, tighter T. And one that releases the ATP, open or O. Each 120-degree turn forces each site into the next state in the sequence. L to T, T to O, O to L.
So one full 360-degree rotation.
Synthesizes and releases three ATP molecules.
Incredible. And people have actually seen this rotation.
They have. Remarkable experiments by researchers like Masasuke Yoshida and Kazuhiko Kinoshita Jr., they attached a fluorescent actin filament to the rotating part and literally watched it spin under a microscope when supplied with ATP. Visual proof of the motor.
Amazing. You also mentioned something about the number of subunits affecting efficiency.
Right, that rotating C ring in the Pho part. The number of C subunits isn't fixed across all species. It can range from 8 in some bacteria up to maybe 17 in others. In mammals, it's typically 8. Since it takes one proton flowing through to advance the ring by one subunit, the number of protons needed for a full 360-degree turn, and thus 3 ATPs, depends on the number of C subunits.
Ah, so if it takes 8 protons for a full turn in mammals, that means 8 protons yield 3 ATP.
Roughly, yes. And that helps explain why the PO ratio of the amount of ATP made per oxygen atom reduced isn't a neat whole number. For NADH, we pump 10 protons, plus we need protons for transport, leading to about 2.5 ATP. For FADH2, which enters at complex 2 and pumps fewer protons, maybe 6, it's about 1.5 ATP. The C-ring stoichiometry is key to this.
Okay, that makes sense. So this proton gradient, it's not just for making ATP then.
Correct. The proton motive force powers other essential transport processes across that inner mitochondrial membrane too. Things that are needed for oxidative phosphorylation to keep running.
Like what?
Well, think about it. ATP is made inside the matrix, but it's needed out in the cytosol. ADP and phosphate are in the cytosol, but needed inside. So you need transporters. The proton motive force helps drive the adenylyl nucleotide translocase, which exchanges ATP out for ADP in, and the phosphate translocase brings phosphate into the matrix along with a proton, using the pH gradient part of the proton motive force. It's all interconnected.
A whole support system powered by the same gradient. Okay. But what about getting fuel into the chain? You mentioned NADH earlier. What about the NADH made during glycolysis out in the cytosol? That can't get through the inner membrane, right?
Excellent point. The inner membrane is impermeable to NADH. So the cell uses specific shuttle systems to get the electrons from cytosolic NADH into the mitochondrial matrix without moving the NADH molecule itself.
Shuttle systems, like passing the electrons to another molecule that can cross.
Exactly. There are two main ones. The maltodaspartate shuttle is predominant in liver, kidney, and heart muscle. It's very efficient. The electrons effectively enter the chain at complex Holm, so you still get about 2.5 ATP per cytosolic NADH.
And the other word?
The other is the glycerol-3 phosphate shuttle, more active in skeletal muscle and brain. It's faster, but less energy efficient. The electrons end up being passed to FAD within the inner membrane, effectively entering at complex 2.
Ah, so those electrons only yield about 1.5 ATP.
Correct. So the ATP yield from glucose depends partly on which shuttle system is primarily used in a given tissue.
Nature finds a way. And speaking of nature's ingenuity, tell me more about those hot plants. That sounded wild.
Oh yeah, the thermogenic plants. It's fascinating. Plants like the eastern skunk cabbage or others in the arum family, they can raise their temperature significantly above the surroundings.
How? By uncoupling, like we discussed.
Precisely, but using their own specialized enzymes. They have alternative respiratory pathways, an alternative NADH dehydrogenase that bypasses complex mine, and crucially, an alternative oxidase, AOX, that accepts electrons from ubiquinone and reduces oxygen to water, but without pumping protons like complex 4 does.
So electrons flow, oxygen is used, but no proton gradient is built up by that step.
Right. The energy from that electron flow is just released as heat. It allows the plant to melt snow in early spring or volatilize smelly compounds to attract pollinators. A really cool metabolic adaptation, bypassing ATP synthesis or heat.
Incredible. Okay, so this whole elaborate system must be tightly regulated, right? The cell wouldn't just run at full blast all the time.
Absolutely not. It's exquisitely controlled, primarily by the cell's immediate need for energy. The main factor is the availability of ADP.
ADP, the substrate for ATP synthase.
Yes. This is called acceptor control of respiration. If ATP levels are high and ADP levels are low, meaning the cell isn't using much energy, ADP becomes limiting for ATP synthase. This slows down proton flow through the enzyme, which causes the proton gradient to build up.
Which then slows down the electron transport chain because it's harder to pump protons against that steeper gradient.
Exactly. It's a beautiful feedback loop. Electron transport, oxygen consumption, and ATP synthesis are all geared to the rate at which ATP is actually being consumed by cellular work. The system essentially self-regulates based on demand. The ratio of ATP to ADP in AMP is a key indicator of the cell's energy status.
So it only makes ATP as fast as it's needed. Makes sense. But what happens if things go wrong? Like what if oxygen runs out? Hypoxia.
Hypoxia. That's a critical situation. If there's no oxygen to accept electrons at the end of the chain, the whole process backs up. Electron transport stops. Proton pumping stops. The proton motive force collapses.
And ATP synthase could then run in reverse, right? Using up precious ATP to pump protons back out.
Potentially, yes. Hydrolyzing ATP would be disastrous when energy is already scarce. So the cell has a safeguard. A natural inhibitory protein called IF1.
IF1.
Yes. Under normal conditions with a healthy proton gradient, IF1 is inactive. But when the proton gradient collapses, Specifically, when the matrix pH rises closer to neutral or slightly alkaline, which happens in hypoxia, IF1 binds tightly to ATP synthase.
And stops it.
It inhibits its ATPase activity. It basically puts the brakes on, preventing it from running backward and wasting ATP. It's a crucial survival mechanism during oxygen deprivation.
Wow, cells have really thought of everything.
Well, evolution is certainly selected for robust mechanisms, and hypoxia triggers broader adaptive responses too. Low oxygen activates a transcription factor called HIF1, hypoxia-inducible factor.
Okay, what does HIF1 do?
Think of it as a master regulator for dealing with low oxygen. It switches on genes that help the cell cope. For example, it can lead to changes that decrease reliance on mitochondrial respiration, promote glycolysis, and even trigger changes in the subunits of the respiratory complexes themselves, like swapping parts of complex 4 to make it work better at very low oxygen concentrations. It's a whole coordinated response.
It really highlights how interconnected everything is. Glycolysis, the citric acid cycle, oxidative phosphorylation, they aren't separate pathways, are they? They're all talking to each other.
Constantly. Regulated by shared signals like ATP-AD ratios, NADH levels, even intermediates like citrate. It's a finely tuned metabolic network designed to maintain energy homeostasis.
And you mentioned earlier that mitochondria do more than just ATP and heat. Steroid synthesis, apoptosis.
Yes, let's revisit that because it's so important. Mitochondria are key sites for synthesizing steroid hormones. They contain specific enzymes, cytochrome P450s, that perform crucial hydroxylation steps in making hormones like cortisol or testosterone.
And apoptosis, programmed cell death. How are mitochondria involved there? It seems counterintuitive for a powerhouse.
It does, but they play a central role. In response to certain death signals, the outer mitochondrial membrane can become permeable to proteins usually kept inside. One key protein is cytochrome C.
Cytochrome C, the electron carrier from the respiratory chain.
The very same one. But when it escapes into the cytosol, it takes on a completely new role. It's a classic example of protein moonlighting.
What does it do in the cytosol?
It triggers the assembly of a complex called the apoptosome. This structure then activates a family of proteases called caspases. And these caspases are the executioners. They systematically dismantle the cell from within. So the release of cytochrome C from mitochondria is often a point of no return for apoptosis.
Mind-blowing. A simple electron carrier becomes a death signal. Okay, one more layer. The genetics. Mitochondria have their own DNA.
They do. A small, circular, double-stranded DNA molecule called mtDNA. It's tiny compared to our nuclear genome. In humans, it only encodes 37 genes.
Only 37. What do they code for?
13 of them code for protein subunits of the respiratory chain complexes, I3, 5, and V. The rest code for ribosomal RNAs and transfer RNAs needed to actually make those 13 proteins right there inside the mitochondria.
So wait, only 13 proteins for the respiratory chain, but there are hundreds, maybe thousands of different proteins in a mitochondrion. Where do the others come from?
From the nucleus. The vast majority, something like 1,200 different mitochondrial proteins, are encoded by genes in our regular nuclear DNA, synthesized on cytosolic ribosomes, and then imported into the mitochondria. It's a dual genetic origin.
Which fits perfectly with that endosymbiosis theory, right? The bacteria brought some genes, but over billions of years, many got transferred to the host nucleus.
Exactly. The mtDNA, the mitochondrial ribosomes, the tRNAs, they're all strong evidence for that ancient bacterial origin. Even the way bacterial flagella work, powered directly by a proton gradient, echoes the chemiosmotic principles we see in mitochondria.
Now this mtDNA, does it have implications for health and disease? Does it mutate?
It does, and that's a critical point. mtDNA tends to accumulate mutations at a higher rate than nuclear DNA.
Why is that?
Several reasons. One is proximity. It's right there on the mitochondrion, ground zero for ROS production from the respiratory chain, which can damage DNA. Also, the DNA repair mechanisms within mitochondria aren't quite as sophisticated or efficient as the nuclear ones.
And what's the consequence of these accumulating mutations?
It's thought to be a major contributor to aging. The gradual decline in mitochondrial function due to accumulated mtDNA damage could explain, at least in part, the age-related weakening we see in tissues with high energy demands, like skeletal muscle and heart muscle.
That's fascinating. And how are these mutations inherited?
Strictly maternally. All your mitochondria come from the egg cell. The sperm contributes virtually none. So mitochondrial DNA and any mutations in it are passed down only from mother to offspring.
And you mentioned heteroplasmy. What's that?
That means that within a single cell or tissue or even organism, there can be a mixture of mitochondria, some with the mutation, some without. The proportion of mutant mtDNA can vary wildly.
Ah, so that explains why mitochondrial diseases can have such variable severity, even in the same family.
Precisely. If you inherit only a small percentage of mutant mtDNA, you might have mild symptoms or none at all. If you inherit a very high percentage, especially in critical tissues like the brain or muscle, the disease can be severe. It depends on the specific mutation, the tissue, and this level of heteroplasmy.
Are there specific diseases linked to mtDNA mutations?
Yes, a group often called mitochondrial encephalomyopathies, because they typically affect the brain, encephalo and muscle myopathy. Things like LHON, lipohereditary optic neuropathy, which causes blindness due to defects usually in complex 1, or MERF syndrome, myoclonic epilepsy, with ragged red fibers often caused by mutation in a mitochondrial tRNA gene, which messes up protein synthesis within the mitochondria.
Ragged red fibers.
Yeah, under a microscope, muscle biopsies from MERF patients often show muscle fibers with abnormal accumulations of mitochondria, which stain red with a particular dye, giving them a ragged appearance. It's a hallmark of mitochondrial dysfunction. But remember, it's not just mtDNA. Mutations in any of those 1,200 nuclear genes encoding mitochondrial proteins can also cause devastating mitochondrial diseases. It's complex.
So, given the maternal inheritance, what options are there for families affected by severe mtDNA diseases?
This leads to a cutting-edge and sometimes controversial area, mitochondrial donation. Techniques sometimes referred to as three-parent IVF, although that term is a bit misleading.
How does that work?
Essentially, you take the nuclear DNA from the intended mother's egg, which carries the mtDNA mutation, and transfer it into a donor egg that has had its own nucleus removed but contains healthy mitochondria. Then you fertilize that reconstructed egg with the father's sperm.
So the resultant child has nuclear DNA from both parents, but mitochondrial DNA from the egg donor.
Correct. It allows a woman with pathogenic mtDNA to have a genetically related child without passing on the mitochondrial disease. It's technically complex and raises ethical discussions, but it's a potential solution for some families.
Really illustrates the importance of these tiny organelles. Any other direct links between mitochondrial health and common diseases?
Oh, absolutely. There's a rare form of diabetes, for example, caused directly by mitochondrial defects in the pancreatic beta cells. These cells need to make a lot of ATP in response to glucose to trigger insulin release. If their mitochondria are faulty due to specific gene defects, they can't make enough ATP, insulin isn't released properly, and you get diabetes. And more broadly, mitochondrial dysfunction and accumulated ROS damage are increasingly implicated in neurodegenerative diseases like Alzheimer's, Parkinson's, Huntington's, as well as in heart failure and the general aging process. They really are central to health and disease.
Wow, what an incredible, intricate picture. We went from the basic idea of a powerhouse to this complex, ancient machine involved in energy, heat, signaling, cell death, genetics, aging, and so many diseases. Oxidative phosphorylation is truly at the heart of so much of biology.
It really is. The mitochondrion is far more than just a furnace. It's a dynamic hub, constantly sensing and responding, deeply integrated into the life of the cell. So, thinking about all this complexity, this adaptability, this central role, what do you think are the next big questions? Where does mitochondrial research go from here? What's the next frontier in understanding or maybe even intervening in these processes?
That's a great question to leave us with. The potential seems huge. We really hope this deep dive into oxidative phosphorylation sparked your curiosity and gave you some valuable insights. Thank you so much for being part of the Last Minute Lecture family. We appreciate you joining us. Until next time, keep digging deeper.